Since everyone's quiet, maybe we should get started. We should take advantage of this.
That's good.
All right, so I'd like to welcome everybody. You guys should sit down. You're supposed to sit and then come up.
There's custom.
There's uproarious applause. So it's my great pleasure to introduce Carson Bryant to give his thesis seminar today. He graduated magna cum laude from Vanderbilt in 2017, where he majored in chemistry and mathematics and minored in scientific computing. He won many awards in chemistry. He entered Yale through the PEB program, and PEB stands for, because I can never remember, Physical and Engineering Biology, right? And somehow he ended up in my laboratory, for which I am very grateful. I have no idea how he found his way. So in my laboratory, he's really brought us into the 21st century by adding to our repertoire a set of skills that are highly based on mathematics, which he's really good at, and data science. And you're going to see a lot of that today. You can see some of it even right here, right? So this is something that he did. He's been incredibly prolific in my laboratory. He's published a number of papers. I actually had to go back and look because I can't keep them all straight. As far as I can tell, he's had 3 co-first author, either first or co-first author papers for original research papers, and 2 middle author papers, and has one currently submitted to nucleic acid research, which is really what he's going to talk about today. So I'm very grateful that Carson was in my laboratory for all these years, and I'm really proud to be able to have him tell you about his PhD work. So without further ado, Carson will tell you about uncovering novel biological regulators of ribosome biogenesis. You can come up.
Yes, thank you so much for the lovely, wonderful introduction. Thank you for being here today, and thanks to all of you for being here today, and our folks on Zoom. Hopefully we're transmitting okay. Yeah, it's my pleasure and my privilege to share a little bit of my work with you today. And I want to start by saying one thing I don't want to do today is reach all my papers. So Susan had a nice, summary of the work that I did, and I don't want to go into all that. Frankly, sometimes at these things people go on and on lots of detail, and frankly, it would just be really egotistical. And just, for me to stand here and say, "Oh, look at all these papers I published. How smart I am. I'm looking for a job. Please, can anyone hire me?" Something like that. So I don't want to do that today. And that would be outrageous, right? So instead, and sometimes when people do that, this is actually what happens to me. This is a fault of my own, but I don't want to make you feel this way today.
This is so boring.
So I'm going to try and avoid doing that to you today. And instead, things that I do want to do today are tell you the most interesting things that I discovered in grad school. I'm going to tell you why they matter, and I want to engage and include you. And so this talk today is going to be, some summary, high-level things. I will unfortunately have to show you some data, so avert your eyes if that makes you ill. But I do want this to be an accessible talk for everyone. Okay, so the roadmap for today is briefly I will tell you about the scientist that I was before grad school. I want to tell you about the ribosome today because the ribosome is really, really cool. In Susan's lab, we study how the ribosome is made, but the ribosome is an amazing machine, and I want to talk about it just for a minute. I also want to talk, yes, I will tell you about how ribosomes are made, and 3 of my projects, which are a way that we can measure ribosome biogenesis, how microRNAs may regulate this process, and I also want to tell you about a rare disease caused by aberrant ribosome perfection. Okay, so this is me, circa 2014. I've got the same hair you've come to know and love, but I'm sporting a different watch. And as Susan wrote in her introduction, yes, I studied chemistry, applied math, and scientific computing. which gave me an engineering mindset. And engineers like to solve problems using existing tools or make their own tools to solve problems. And so two of the things that I was interested in before grad school were one, making photosystem 1 biosolar devices. And so I worked in Dave Koval's lab at Vanderbilt, and essentially Gary Redwig also works on this problem here at Yale. But basically you can take this huge plant protein complex that harvests light energy, and you can put it to use by in biosolar cells and make better, greener energy collection materials. So I thought that was really cool, using proteins to do a job. I was also really interested in protein structure prediction and protein design, especially the latter, because that's where we get to make our own tools, right? And this field has come a very long way since I entered grad school with the advent of deep learning. But essentially, these are both, questions about using proteins to do some kind of job. What I haven't thought much about yet, before I came to grad school, were things like, how are proteins made? How are the protein makers made? How is this all regulated? How do these processes tie into being a cell? And how do problems therein affect medical outcomes? And I had the heretical question, is RNA anything besides a boring intermediate? So I'm very privileged to come and learn answers to all these questions here at Yale. But as I was writing my thesis, my intro to my thesis, I actually I was reflecting on how much I learned on these topics, and there was one character who came to mind, and that was LUCA. Oh, wait a second, not that LUCA, this LUCA, the last universal common ancestor. So LUCA lived 4 billion years ago, and was ineligible to play for the Dallas Mavericks, which were founded in 1980. But LUCA was the MVP on primordial Earth, as you can see there. And that's because LUCA had the skills to pay the bills, as the kids would say. So LUCA had already developed ways to extract nutrients from its environment, extract energy, and something near and dear to many people in this room is the central dogma of molecular biology, which was already well enshrined in LUCA, even 4 billion years ago. And you can see the ribosomes are part of this. And so this is a simplified diagram of the central dogma. And put simply, the central dogma says that DNA codes for RNA, which then codes for protein. And a classical, bit outdated way to think about this is that DNA and RNA, which are biopolymers of nucleic acids, are informational molecules, and then they get translated into a functional molecule called protein, which is a biopolymer of amino acids. And this, my engineering mindset, makes me also sometimes think of this as a digital to analog conversion. Although I know all three of these molecules are physical and have their own functions in the cell. But that means if translation is the central process of the central dogma, then the main character is the translator, or the ribosome. I'll show up. The ribosome. And so this translator, the ribosome, says, hey, DNA, RNA, I hear what you're saying. That's great. But now it's time for something completely different. And it translates a protein. The ribosome itself is actually composed of two different subunits, the large 60S subunit in eukaryotes, and the small 40S subunit. And it's actually composed of ribosomal RNAs and ribosomal proteins. And for once in biology, they're not trying to trick you with the name. It's just what it says on the tin. But I want to call out this green speck here in the middle of the large 60S subunit. That's actually where proteins are made. And so for a long time, this was suspected to only require ribosomal RNA, but it hadn't yet been unequivocally proven. And so I'm telling you this because I want to set you up for perhaps the greatest 2 slides in Nobel history. The question again is rRNA responsible for protein synthesis? And it was in the year 2000 that Tom Seitz's group here at Yale actually discovered that they found high resolution structures of the ribosome, and they found that these ribosomal proteins are very far away from where the site where proteins are made. And so that means that Tom Seitz's group was able to answer this question with a resounding yes. rRNA is responsible for protein synthesis. And that's pretty remarkable. This fact is so remarkable, if you leave today and you only remember one thing, I want it to be this. And so Tom's next slide was for if you were taking a big snooze and you missed the big news. He had a sense of humor. And he said, the ribosome is a ribozyme. Wow, amazing. So to remember this, we should all chant it together. So when I say ribo, you say zome. Can we do that?
Yeah.
Ribo, zome. Ribo, zome. When I say ribo, you say zyme. Ribo, zyme. Ribo, zyme. Very good. You want to do it again? Let's go again. Ribosome. Ribosome is a ribozyme. Ribozyme. Okay. And so this is an incredible fact. I mean, really, please do never forget this for the rest of your life. It's amazing. It's amazing. And I didn't know this before. This is part of the reason it's so exciting to study RNA, to study ribosome, to study how ribosomes are made. Here we go. Okay. So the ribosome is this amazing RNA machine. But this leads us to the next question, which I'm sure all of you are thinking. Quis custodia ipsos custodius? And of course, this has many meanings in Latin, but I would translate it to be who will manufacture the manufacturers. How are ribosomes actually made themselves? And this is the process that we study in Susan's lab. It's called ribosome biogenesis. In eukaryotes, it begins in the eukaryotic nucleus, which is a membraneless, phase-separated organelle. And this is already a highly simplified diagram, but I'm going to distill this into 3 simple steps for you today, even further. So the first step of ribosome biogenesis is step 1, transcription of the pre-ribosomal RNA by RNA polymerase 1. Remember, our RNA is actually what makes protein. So in some ways, this is actually the most important step. It's the first step. It's very important. And so you can see that this 47S pre-rRNA transcript is transcribed by RNA polymerase 1. Great job. The transcript will contain 3 of what will become 4 of the mature ribosomal RNAs in the cell, in the ribosome. But what you can see is actually there's these non-functional transcribed spacer regions linking these what will become these mature rRNAs. And so we have to get rid of those, right? And so the next step is processing of the pre-ribosomal RNA and some other stuff. But let's just say processing. So things that happen in step 2 are we get rid of the transcribed spacer regions. The pre-RNA is modified so it can become methylated and pseudo-uridylated at different positions. The 5S RNA is transcribed in the nucleus and that gets put into the mature ribosome. But this is highly simplified. In the cell, this is a essentially organized chaos. Okay, or as my lab might say, curated chaos, because these transcripts are being bound by over 80 permanently installed ribosomal proteins, and they need the help of over 200 transacting assembly factors. So this is I can't possibly show you in one slide how complex this is, but a lot of work has been done to capture structural models of how the ribosome is built in cells by cryo-electron microscopy. So that's great. So that's step 2, processing. And then we have the ultimate output of ribosome biogenesis, which is actually making proteins, right? That's the whole point of making the ribosome, is then to go and make a protein. So active protein synthesis is the final step. So just in case you missed it, step 1 is transcription, step 2 is processing and some other stuff, and step 3 is protein synthesis. Great. Okay, so who cares? Well, it's important to study ribosome biogenesis biologically for one reason. So a lot of cellular attention is devoted to controlling the process of making ribosomes. Most transcripts in the cell are pre-ribosomal RNA. Most mRNAs code for RPs or AFs. And ribosome biogenesis is also very tightly co-regulated with the cell cycle. So therefore, controlling ribosome biogenesis is a main objective of the cell. And it's important to study ribosome biogenesis from this perspective because it teaches us about cellular strategies for growth, for construction of huge 3D molecular, macromolecular complexes, and for about how the cell chooses to use its energy efficiently. It's also important to study ribosome biogenesis clinically. And so the first reason is that problems with ribosome biogenesis can cause rare diseases known as ribosomopathies. The most infamous ribosomopathies are a class of bone marrow failures, and that's really bad because it means you can't make enough red blood cells or white blood cells. There's another class of ribosomopathies that affect development, specifically development of important organs like the brain or the heart or the skin and hair, or development of the face. These are really important things, right? And so it's really important to understand what's going wrong, how ribosome biogenesis is going wrong in these diseases. It's also important to understand ribosome biogenesis from an oncology perspective, because it's the case that hyperactive ribosome biogenesis or, interestingly, impaired ribosome biogenesis can each in their own way lead to oncogenesis and cancer progression. And so it's important to understand ribosome biogenesis clinically because it tells us how to better diagnose, treat, and ideally prevent human diseases. So what I've told you so far is that ribosome biogenesis is complex. I distilled it into 3 main steps for you: transcription, processing, and some other stuff, and protein synthesis. It's also conserved all the way back to LUCA 4 billion years ago. And in eukaryotes, it became a little more complicated, both the structure of the ribosome itself, as well as how the process of ribosome biogenesis. And it's also compelling to study ribosome biogenesis because this can help us understand biology, which is important, but it can also help us treat human disease. That's great. And despite all the things that we know about ribosome biogenesis, there are still a lot of unanswered questions in our field. And so today I want to tell you about some of my specific contributions to this field. First, I want to tell you a little bit about how microRNAs may be regulating this process. And en route, we will discuss a high-throughput method for measuring RB. And so first, I want to thank the team who helped answer these questions. Thanks to Mason, my dedicated, devoted baymate and friend for all his work on these projects. I want to thank Gabriella, who is a very talented MD PhD rotation student in our lab. And I want to thank our lovely collaborators at the Yale Center for Molecular Discovery, Yulia Sorosteva and Laura Abriola. Okay, so in 2020, Mason, Susan, and I wrote a review on how non-coding RNAs represent an emerging higher order regulatory layer governing ribosome biogenesis. And so specifically, we looked through the literature, Mason and I, for examples of microRNAs or long non-coding RNAs, LINC-RNAs, that are already known to regulate some of the 3 steps in ribosome biogenesis. You can see in this diagram: transcription, processing, some other stuff, and protein synthesis. And microRNAs were particularly compelling to me because microRNAs are actually already known to regulate really complicated processes like cancer and development and metabolism, things that are very hard to control and very complex. So engineering mindset, if you have a tool in the Swiss Army knife that can already do this, maybe there are other tools out there that people haven't appreciated yet that can regulate ribosome biogenesis. Furthermore, the answer to that question already is yes, right? I was able to find a few examples of these in the literature. So the question that I wondered from this review is what other microRNAs could be functioning as ribosome biogenesis methods of control. And so to do this, I used a nucleolin number assay, but first I want to tell you that nucleolin number in cells is cell type dependent. And so in our lab, we use human MCF10A breast epithelial cells. They're non-cancerous and have a nearly normal karyotype, which is why we use them, because they're almost normal. And these cells, when they're in their happy place, they have most often between 2 and 4 nucleoli. Now, an interesting discovery from our lab is that if you treat these cells, if you manipulate them somehow, if you do some genetic manipulation or if you give them a drug, and you observe a change in nucleolin number, that's a very strong predictor of interrupted ribosome biogenesis. And our lab was able to use this to great effect in multiple publications. So Katie Farley-Barnes and Kat McCann in Susan's lab discovered 139 novel protein regulators of ribosome biogenesis that caused, upon their depletion, caused a decrease in nucleolin number. And Lisa Ogawa-McLean would call this the fried egg phenotype, because it looks like you're frying an egg. On the other side, Lisa herself discovered 113 novel protein regulators of ribosome biogenesis that caused what we call the 5+ nucleoli phenotype, or the starry egg. And so together, these 2 manuscripts led to the discovery of over 250 novel protein regulators of ribosome biogenesis. Amazing. So I saw this, and I was thinking about my question about microRNAs, and I said, let's do this for microRNAs. Right? You have a tool already in the engineering toolbox, let's use it again. And so what I did, to quickly summarize, is I screened a library of 2,603 human mature microRNA mimics. to ask this question: are there microRNAs that are could be interrupting ribosome biogenesis? And the answer is yes. Actually, the images I've been showing you all along are from my work. I got you. So, just kidding. This middle panel indeed is the negative control for our assay, where most cells have between 2 and 4 nucleoli. And then on the left and the right, I'm showing you the top hits that I found of microRNA mimics that, when added to the cells, cause a change in nucleolar number. Of course, I did this in high throughput, so I was actually able to identify 73 high confidence microRNA hits in total using this high content nucleolar number screen. And most of these hits were 1 nucleolus hits, Rib eggs, but I did find a couple handfuls of 5+ nucleoli hits. Those are the starting eggs. And 71 of these 73 hits passed hit-pick validation. And I would love to tell you more about these double flashlight plots if you want to ask me later, but I have to move on. And so this is great. So these are putative, novel, negative regulators of ribosome biogenesis. But the question then is, are these microRNA hits actually having a direct impact on ribosome biogenesis? Because the nucleolar number screen is not very direct. It's sort of indirect. And it's helpful, but it's not directly telling us about RB. And so for this, Mason and I actually teamed up and improved and updated the definition of an assay that's been used by our field to measure RB. And so the way this assay works is, much like in the primary screen, we can use our MCF10A cells and reverse transfect them with siRNA controls or microRNA mimics. There we go. For 72 hours. And then this is where the magic happens. So in the last hour that the cells are alive, we treat them with this wonder molecule called 5-ethynyluridine, or 5-EU. And this is 5-EU here. 5-EU is essentially a building block of RNA. It's an analog of uridine, which is one of the 4 RNA bases. And so you can feed cells with 5-EU, and they'll actually put it into all the new RNAs that they make. But 5-EU has a trick up its sleeve. So 5-EU has this other alkyne moiety, and that essentially allows researchers like me and people in my lab to go into cells and access only the new RNA that's been synthesized during the treatment period. And since most new RNA in the cell is pre-ribosomal RNA, this is a really easy way to directly assess how ribosome biogenesis is functioning. And so for this assay, we do some other stuff. We look at nucleolid 5-EU, blah, 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 not important. Paper. But I do want to show you what it looks like when we actually access the new RNAs. And so in happy cells, again, you can see most the new RNA in the cell is pre-ribosomal RNA, so you can see bright foci that represent the nucleoli. However, when you knock down something that's very important for step 1, which is polar RNA, a subunit of RNA polymerase 1, you can see that the light that was once brightly emanating from the nucleolus has been extinguished. And that's because the cell can no longer make the same amount of pre-ribosomal RNA. So using this assay, which took a while, I also want to say, just to show you, because I don't have all the time in the world to talk about me and Mason's work. Like I said, our work improved and more clearly defined this assay for measuring ribosome biogenesis. So we improved this assay by optimizing it for high throughput, by making sure that we can measure only nucleolar 5-EU when able, and that makes sure that we're cutting down background from pesky mRNAs and tRNAs in the nucleoplasm. It also uses a defined pipeline for the free open source software cell profiler to make the image quantification more reproducible. And I think Mason's contribution to this work is actually probably the most important, is that this allowed us to clarify what this assay actually measures. People thought that it just measured step 1, but it measures step 1 and step 2 and some other things. Okay. So that's an important clarification that I won't bore you with more than that. Okay, so we did all this work to get this assay, blah, blah, blah. Okay, what does it look like when you look at the hits? Okay, so strikingly, most of these microRNA hits are strongly inhibiting nuclear RNA biogenesis. And so you can see in this panel, these are our happy cells where lots of new RNA is being made, especially in the nucleolus. And in these other panels with all these microRNA hits, you can see that the nucleoli are essentially indistinguishable in most cases from the nucleoplasm. So that means that ribosome biogenesis is being directly inhibited. And of course, we can quantify this, and indeed, most of these microRNA hits are strongly inhibiting nuclear RNA biogenesis, once again, just by looking at this. So this gave me a lot more confidence that these hits are real. They're really inhibiting ribosome biogenesis. I did some bioinformatics analyses to essentially ask the question, why these hits? Why did we find these ones? And essentially what these analyses support is that these microRNA hits preferentially target mRNAs, which encode nucleolar proteins, which are probably going to be important for ribosome biogenesis. And they also target cell cycle factor mRNAs. And as I told you earlier in the intro, the cell cycle is very tightly intertwined with ribosome biogenesis. So if you affect the cell cycle, it's likely that you're affecting ribosome biogenesis too. So then the question became, how is a subset of these microRNAs affecting our three main steps: transcription, processing, and global protein synthesis? Step one, I'll just tell you, it doesn't seem like these microRNAs are really affecting step one. So then we move to step two, and so the question is, how did these microRNA mimics affect step two, pre-RNA processing? And this was a very compelling question for me, because no microRNAs had yet been found to directly affect pre-RNA processing. Indeed, there's a big hole on the side of the diagram that Mason and I drew on the microRNA side by step two processing. And so pre-RNA processing, just to give you a bit more detail, it happens in a stepwise fashion. So remember, the goal of this is to excise these non-functional transcript spacer regions so that we can liberate the mature rRNAs for our mature ribosome. And it happens in this stepwise fashion, and you can actually monitor how this is happening using an experiment called a Northern blot. And when you do that, you can see the cell has a normal pre-RNA processing pattern. So you can see a nice band for the 47S/45S, nice band for the 30S, nice band for the 21S. And these other two bands are also visible. But this is what happens in normal cells. So then the question is, when I treat cells with my microRNA hits, does this change? And oh boy, does it. A subset of these microRNA hits drastically alters human pre-RNA processing. Even if you didn't understand a word I just said before all this, look at this block, okay? This is normal, this is not normal, right? The 30S band is up, it's down, it's all around town. And so your eyes are not deceiving you here, and I've replicated this. And indeed, when I quantify these results, a subset of microRNA hits was shown to dysregulate specifically 30S processing levels. And so this column, I know this is confusing, but if we just look at this column, this is the 30S levels. And so there's three main clusters here. One where levels of the 30S are not affected. That's for a few of these microRNA hits. There's a big cluster where 30S levels are down, as you saw. And then there's another cluster where 30S levels are up at the expense of the product, the 21S precursor. And this is another way to show those results, these clusters. One other thing that I noticed is that for these bottom three microRNA hits, 21S levels are very severely affected. And that's important because the 21S precursor is essentially a direct progenitor of the mature 18S rRNA. So you would expect, if the 21S is really, really down, that 18S levels would also be severely impaired, which is a very severe defect because that means you can't make enough small subunit ribosomal subunits. And indeed, when I checked this, in fact, this is totally consistent. MIR28, MIR708, and MIR4730 showed a decrease in mature 18S levels. So step two, heavily inhibited by microRNA hits. Revelation, right? This was not known to occur before. And then that leaves step three. How was step three, protein synthesis, affected by these microRNA hits? And so we can look at protein synthesis using an experiment called the puromycin incorporation assay. Details, not that important for today. But you can look, when cells are happy, they make a lot of new protein, right? And so that means that there's essentially a lot of new protein being made. Yes, I just said that. And you can see that on the plot. However, when you knock down ribosome biogenesis factor called NOL11, UTP8, or if you knock down ribosomal protein, the cells can't make enough ribosomes anymore, right? And so their capacity for making new proteins is diminished. So that means you get less new protein. So the question I had then is simply, if I treat cells with these microRNA hits, does it affect step three, protein synthesis. And indeed it does, which is exactly what you would expect if these hits are affecting ribosome biogenesis. Almost all of these hits are strongly abrogating protein synthesis, except this one, and it's a nice visual readout. Okay, so for this part, what I've told you is that these microRNA mimic hits are inhibiting the ribosome biogenesis subprocesses of step 2, processing, which was unexpected, and step 3, protein synthesis. For the biologists in the room, I will just quickly say, these microRNA hits dysregulated TP53 levels, which is interesting. Sometimes it went up, sometimes it went down. But almost all of the hits actually upregulated levels of CDK and LUNA, P21, which is an important factor for arresting the cell cycle during times of cellular stress. And a little inside baseball here, these two microRNAs here, 28 and 708, are family members, and the only reason that they don't upregulate P21 is because they actually directly target P21, which is unexpected, but came out of some of the other figures in this manuscript. Okay, so why is this all important? Well, it's important, of course, to understand how biology is regulated, how biology may regulate itself. But remember, I have an engineering mindset, so I want to know how can we put this to use. And so it's true that currently small molecules are being used as chemotherapies to target the nucleolus and ribosome biogenesis. Remember, I told you that hyperactive RB leads to oncogenesis and cancer progression. So it's very in vogue now to target ribosome biogenesis as a way to treat people's cancer. What I envision is that these newly appreciated microRNA mimic inhibitors of ribosome biogenesis could be used on their own or wholesale or in part of a cocktail, a drug cocktail with to target the nucleolus in oncology. And it is also important to know that actually many of the microRNAs that I discovered are actually already known to be tumor suppressors. And so with this additional information of how these microRNAs are affecting ribosome biogenesis, I think that my hope is that my work will accelerate development of microRNA therapeutics for cancer, and that the biotech and pharma oncology space will more readily embrace these microRNA therapeutics in development. Okay. So now I want to tell you quickly about a second scientific contribution of mine, I guess a third scientific contribution of mine to the ribosome biogenesis field, which is a novel discovery of a novel molecular basis for the rare ribosomopathy, A&E syndrome. And so again, I want to thank the team that helped make this happen. This is an international team of clinicians and researchers. Many of them are Brazilian, and our bedside to bench approach was key in defining this novel molecular basis for A&E syndrome. And it was a pleasure to work with these folks, and it was a privilege to help lead this international collaboration across hemispheres and scientific disciplines. And I also want to thank the patient and the patient's family for allowing us to share their story with the scientific community. Okay, so what is A&E syndrome? A&E syndrome stands for alopecia, neurologic defects, and endocrinopathy syndrome. And it's a rare ribosomopathy. Before this work I'm going to tell you about today, there was one case report that reported 5 adult male siblings who had consanguineous parents. So essentially that means their parents were blood related, which is unfortunately not great for genetics. And all 5 of these patients had differing degrees of hair loss, alopecia, and craniofacial defects, like hypodontia and dental caries. They had moderate to severe intellectual disability, and they had hormonal insufficiencies. So that's the endocrinopathy part. These researchers found that all patients had a homozygous L to P variant in the third RRM domain of RBM28. And RBM28 is an assembly factor that helps make the large subunit of the ribosome. Combined with this, all patients were found to have fewer free cytoplasmic ribosomes compared to healthy controls. And so these last 2 facts is what led these researchers to classify A&E syndrome as a ribosomopathy. Just to tell you a bit about RBM28, so this human protein is called RBM28. It also has a yeast ortholog, or a closely related, closely evolutionary related protein called NOP4. Okay? And And both of these, remember I told you, ribosome biogenesis is conserved, right, all the way back to LUCA 4 billion years ago, but especially back to yeast, which are our simple eukaryotes. And RBM28 is an essential large subunit assembly factor, so it's important for making large subunits. It's been most studied in yeast as NOP4. In the mid-1990s, Sun and Wolford discovered that NOP4 is essential for growth, and of course LSU biogenesis. Kat McCann, who was a PhD, graduated from Susan's lab, studied NOP4 more in depth, and she discovered that these third and fourth RN domains, which normally bind RNA, actually are protein binding domains, so they're critical for binding other large subunit assembly factors. And she also found that inserting the classical L-P A syndrome variant in NOP4 disrupts its ability to assist with pre-60S pre-RNA processing for step 2. For our friends who studied the nematode Phaneropetes elegans, RBM28 was found to be important. And if you deplete RBM28, it results in defects in developmental timing and morphology. And of course, in humans, it's known to be essential, and depleting RBM28 causes a similar defect in step 2 pre-RNA processing. Okay, so where do we come in? So our collaborators in Brazil identified a second putative case of AME syndrome. In contrast to the original case, which was 5 adult male brothers that had consanguineous parents, this was a female pediatric patient and with no relation to the original cases. The patient presented with clinical features consistent with AME syndrome, including alopecia, craniofacial defects, including a brachial cleft cyst here, intellectual disability. Brain scans revealed the patient had white matter lesions and calcifications, as well as narrowing of the cerebral artery, and also had dental caries. However, so that's consistent with the A and the N of AME syndrome. This patient was not observed to have hormonal insufficiency, but that when our clinician friends, collaborators thought that this might be the case because this was a pediatric patient, quite young. And I'm sure all of you remember, there's a lot of hormonal changes that happen to you when you go through puberty, right? And so it's possible that as this patient ages, there may be a hormonal insufficiency that arise. Okay, so we wondered, does this patient have the classical LDP variant in RM3 of RBM28? And the answer to this question is a resounding no. Instead, this patient has novel biallelic RBM28 variants. And so you may remember from early in your life that you learned that for most genes in your body, you inherit one copy from your biological mother, one copy from your biological father. And so what the word biallelic means is actually this patient unfortunately inherited essentially one bad copy from their mother, biological mother, and a different bad copy from their biological father. And this is different, of course, from the classical variant because RM3 is not directly affected. And so what do these variants actually do to the output of the protein? Well, the clinicians thought that these might be causing a change in the protein sequence of this protein of RBM28. But in fact, Susan and I quickly looked at these and realized that they're splicing variants. And for instance, you can see here in this panel, the human splice site, 5' splice donor, has a consensus sequence. This is what it should look like, or it looks like most often. And you can see here, this GT is always there. The bigger the letters, the more important it is that that's what the bases are. And that's, of course, in the wild type protein, but in the patient variant for exon 5, that is completely ablated. So that's going to be very detrimental to splicing. And there's a similar story for exon 8 over here. This highly conserved G, which is a G in the wild type sequence, is mutated to a T. And indeed, in silico analysis predict, 4 different in silico analyses predict that these will strongly impair splicing. Then the question is, splicing is complicated. How is it actually going to affect the output of the protein? Is it going to skip the exon? Is it going to activate a cryptic splice site? What's going to happen? Thankfully, the answer was very simple. Indeed, these variants simply cause exon skipping of the affected exon. And so normally during splicing, exon 4 ligated directly to exon 5 and 6. If if you have the patient variant, exon 5 is simply skipped, and you have, unfortunately, exon 4 is spliced directly to exon 6. And it's the exact same story in for the exon 8 variant. And these are in cellular mini-gene splicing assays. Unfortunately, we were not able to get patient samples to actually super duper verify this, but I'm pretty confident in this result. Okay, so what does that actually mean for the output of the protein? Well, if you delete exon 5, it's not the end of the world. It's not great news, but it's not wonderful news. And so if you delete exon 5, essentially you end up with an in-frame deletion of 31 amino acids from the C-terminal end of the second RRM domain in RBM28. And this is probably going to impair RNA binding, because these are two RRM domains are thought to be important for RNA binding. And that will probably impair the ability of RBM28 to make large subunits. So we predicted quickly after getting those last results that the delta E5 allele would be partially functional or hypomorphic. The story is a little worse for the exon 8 variant. So if you skip exon 8, unfortunately, this is your 2 out of 3 chances to have an out-of-frame deletion, and there's quickly generates a downstream premature termination codon. So this is bad news for a couple of reasons. One, it's likely that this allele produces no protein at all, because if you have a premature termination codon, there's this process called nonsense-mediated decay, which can essentially just shred these. The ribosome can detect, actually, that's one of its other functions. It can detect that this is a bad transcript, and it can essentially just say, we need to get rid of this. However, even if any protein is made from this allele, it's going to be missing these crucial RM3 and RM4 domains, which, as Kevin previously showed, are important for interacting with other large subunit assembly factors. And so, with those two strikes against it, it was our prediction that the delta E8 allele would be completely non-functional or null. And so I used a yeast model of disease to test this, these hypotheses. And essentially, one nice thing you can do in yeast is you can essentially just turn off the copy of NOP4 that yeast have, and you can resupply an additional copy. So you can resupply it with wild type normal copy, or you can resupply it with the patient variants. And that will allow you to see the degree of functionality of the patient variants. So that's what I did. I made the patient variants with the classical LDP, as well as the two, the exon 8 and the exon 5 variants. Put them in a NOP4, the endogenous yeast protein, and the human protein as well. And you don't have time to show you the data for this, but you can ask me later. It was clear from these experiments that the patient variants impaired growth and also inhibited pre-60S pre-rRNA processing. Okay, and so this essentially confirmed, strongly supported our original hypothesis that the delta E5 variant was hypomorphic or partially functional, and the delta E8 variant was completely null. And so this work was important because it, one, it defined a new molecular basis for this very rare ribosomopathy, A&E syndrome. It was also important in the context of ribosomopathies more generally. And so many of these diseases of ribosome biogenesis are haploinsufficiencies. And what that means is essentially they affect proteins that are very, very important. And so these diseases like Diamond-Blackfan anemia or Treacher-Collins syndrome affect proteins that are either core ribosomal proteins that have to go in every single ribosome that the cell makes. So it's really important. Or they affect, Treacher-Collins affects TCOF1 treacle, which is a factor that's very important for step 1, transcription of the pre-ribosomal RNA. And so you have to have 2 alleles worth of function in order to not have these diseases. However, our work clarified a couple things. One, it clarified that A&E syndrome is not a haploinsufficiency. It really wasn't clear from the original case report if that was true. But due to whole exome sequencing, trio sequencing, we sequenced both the parents, and it's clear from those results that this is not a haploinsufficiency. Instead, A&E syndrome joins a growing class of recessive assembly factor ribosomopathies, including Schwachman-Diamond syndrome and Bone-Conditis syndrome. And the way that I like to think about this is the following. So if you're trying to build a house, you want to make sure your building materials are really good. If you're trying to build a house with rotten 2x4s that were left outside in the Connecticut winter, you're not going to build a very strong house. However, if the issue with your construction project is that you have a worker who is sick sometimes, or they need to take some time off for family, but they can still get their job done if they're there half the time, then it might be okay. But you're going to run into real issues if you have if that worker is absent for a large period of time, right? And so, yeah, that's how I like to think about these the differences in these diseases. Okay, so what I've told you today is that ribosome biogenesis is complex. I distilled it in 3 steps for you. It's conserved all the way back to LUCA 4 billion years ago, and indeed in our LUCA dumpsters today. And it's also compelling to study because, of course, it's important to understand the biological workings of cells, what make us people, but it's also important to study these because defects in ribosome biogenesis can cause ribosomopathies and cancer. And the work I told you about today, me and my collaborators, really advanced the field of ribosome biogenesis in 3 key areas. So first, my work with Mason and our friends at the YCMD improved and optimized a high throughput assay for directly measuring ribosome biogenesis. And so that advanced the field in the area of methodology. Second, in the area of basic science, I told you how I discovered novel microRNA negative regulators of ribosome biogenesis and how surprisingly they affect step 2 of ribosome biogenesis, pre-rRNA processing. And finally, I told you about how my work advanced the field in the area of clinical molecular pathology. Again, our work with our Brazilian collaborators helped define a novel molecular basis for AME syndrome and clarified the context of this, how this disease fits into the broader context of ribosomopathies. And so I want to thank you all for being here today. It's been lovely to share some of my work with you. You've been a great crowd. Thanks for all the laughs. I want to thank, I have, of course, informal acknowledgements later, but I do want to, of course, acknowledge my lab, my trusting and wonderful mentor, Susan Basurga, is here today. I want to shout out Mason specifically for all the wonderful collaborations we've been on together. Everyone else in the lab, thank you. I want to thank my thesis committee, Dr. Sykes and Dr. Palesti. Thank you for being here. And of course, I want to thank my collaborators at the YCMD. Thanks, Gabriella, and thanks to our collaborators in Brazil, and thanks to our funding. And there are some things that you might still be wondering. And it would be clear, actually, any individual who asked one of these questions, it would be clear you were completely engaged for the entire talk, and that you had thought critically about my work. It would be an honor to answer any of these questions, or any other questions that you may have. So, thank you very much. Happy to hear questions.
I got the one on the top left.
Yes, you have. Why does the nuclear number assay work? It's a mystery, John. Not even you can figure it out. Despite sorry, I didn't mean that. John is brilliant.
You
John is
have
brilliant.
some thoughts about it.
I did, yeah, I sure I have thoughts.
Tell us some thoughts.
So, yeah, the nuclear number assay is very strange. Okay, if you were a venture capitalist and I was trying to sell this assay to you, I would say the nuclear number assay is a amazing, evolutionarily tuned, highly precise classifier of ribosome biogenesis factors, right? Now the recall is not so great, but essentially what that means is we don't know when it works, but it works. Right? That it's essentially a machine learning algorithm that is happening in cells that says it can essentially tell you if you knock something down or if you throw a drug on it, hey, I'm not happy. Now the other thing to note is that in very few cases, only in MCF10A cells, and also Tom Moss's lab has showed in mouse and embryonic fibroblast MET cells, whatever that stands for, Dasha might know, that only in those 2 cell lines that I know of does nuclear number change when ribosome biogenesis is inhibited. And that's sort of a bigger mystery. I think Katie and Kat and Susan wrote in their original paper that established this screen that MCF-taints are special, and they have some like stem cell factors that they express unexpectedly, like SOX2 and NOT4 and some other things. Maybe that's part of it, but I don't know that anyone's looked at,
It's very specific for a small number of cell types.
It seems to be. It seems to be, yeah. Yeah, which is another question that's no one knows the answer to. Ty in my lab was trying to do answer this question, and it was very difficult. And the cells don't really like to be, right? Ribosome biogenesis is essential, so they're already very unhappy if you're trying to inhibit ribosome biogenesis. And then if you're trying to look at them further and really get in there and look at things, cells die very quickly, which I think was a big issue that Ty faced. But yeah, we don't know, but it works, right? Engineering mindset, baby. You don't care why. It just works. Thank you. Madison.
Can I ask you a question that's not on the slide?
You go first.
I ran out of water, so I can't throw it. So I'm gonna do this instead.
Okay.
My question is, so you showed this really interesting P53 data, and you also at some point mentioned kind of offhandedly that you can have like either sort of overactive ribi or underactive ribi, and those are related to oncogenesis. Is that right? Is there a relationship between those two things?
Absolutely. Yeah, so Madison, for Zoom, sorry, I didn't repeat Jen's question, but for Zoom, Madison's question is there a relationship between TP53 levels, regulation, and oncogenesis, essentially? And absolutely there is. Something I completely omitted is that if you interrupt ribosome biogenesis, it triggers something called the nuclear stress response. And so essentially what happens here is RPL5 and RPL11 will go and sequester MDM2, which is the ligase for TP53, blah, blah, blah. And so essentially this activates TP53 levels. Now, another fact is that, especially for diamond blackfan anemia, for the blood diseases, ribosomopathies, of course you have problems making blood cells, which is bad. But these diseases actually predispose you more heavily toward developing cancer later in your life if you're one of these patients. People are actively researching why that is. Alan Warren and others in our field have been looking at that. But yes, it is. They are related. It's the most that I can say right now.
Yeah, so I'm assuming for all your microRNA hits that the primary target is a protein. Do you know that, or are you open to the possibility that it's the RNA itself?
Yeah. So for Zoom,
The ribosomal RNA itself.
Yeah. No. So I actually did. Yeah, it doesn't like you. I got a lot of extra slides here. Well, so I did some bioinformatic analyses to check that, and essentially what I found is that I did a blast search essentially of these microRNA hits on the 47S and said how many predicted sites are there for these microRNAs to bind on the 47S to the primary transcript, and some of the microRNAs, a couple of them had a high number, most of them had a low number or 0, and yet they're still able to directly inhibit ribosome biogenesis, actually in fact to levels that would suggest that they're inhibiting transcription, but indeed they seem to not be inhibiting transcription. And so essentially what I'm saying is there's not a correlation between how readily a microRNA is predicted to bind to the 47S and how harshly it inhibits ribosome biogenesis.
So then what's your interpretation? You think it's still a direct effect, or do you think it's a downstream sort of feedback?
So the other bioinformatic analyses I did, so I did some other bioinformatics. This is sort of on the left. This is a list of what I did, but essentially what I found is that first of all, the microRNAs which were hits are more likely to target proteins that were nuclear, or that's what this one is. B panel is showing. And they're also enriched for targeting things like, I may not be able to read this, but the Go analysis showed that they are enriched for targets involved in the cell cycle and G1 to S transition, and down here for proteins that are localized within the nucleus. So we do think that the primary effect is through targeting nuclear proteins or cell cycle factors or some other proteins that have a reverberation to hit ribosome biogenesis. We don't think it's a direct effect in most cases, actually targeting the pre-ribosomal RNA or other factors like that.
Some of them do target pre-ribosomal RNA.
It's possible. It's possible, but
That's just
there has been work by Keith Giles' lab in UAB, when he had a lab, that showed that microRNAs are tethered to the pre-ribosomal RNA precursor via AGO2, which is essentially saying that they're targeting there. But it's not clear if they're really down-regulating pre-ribosomal RNA transcript directly. Very, very interesting work, and I wish he still had a lab so he could answer this. Hi.
Can you remind me if you did any RNA sequencing or proteomics?
Yeah, in fact I did. So slides in. So I did do some RNA sequencing. And then I'll just share my screen. Last. Okay, so I did do some RNA sequencing for specifically 2 of the microRNAs. And what I found is very interesting. And I would love to tell you about it. Okay, here we go. So actually I mentioned these 2 microRNAs earlier, miR28 and miR708, and they both cause this really severe 21S defect. And that's actually, I found, because they have the same seed sequence. And so they're in the same family, they're siblings, and the seed sequence is essentially what determines what a microRNA targets, right? And so I thought, we could probably figure out why this effect is happening using RNA sequencing, as this ties suggests. And so I did that, and what I found is that by treating cells with either of these microRNA mimics, they generate essentially the exact same transcriptomic profile. And what I mean by that is, so each of these dots is a transcript, a different gene. And when you treat cells, you can see how these gene levels change, transcript levels change. And what you would expect, remember, engineering mindset, applied math, a perfect correlation is y equals 0 plus 1x, right? Look at that equation. What does that say? Y equals 0 plus 1x. So these are doing essentially the exact same thing in cells. And so, the R squared, not too bad for biology, I will say myself. And so specifically what I found is that there are a couple of targets that are really strongly downregulated. SPR3 is one that actually I went on to later find affects SET1 transcription. But there's this other protein, this small subunit ribosomal protein called RPS28. And RPS28 is really important because it's actually already shown to, when you deplete it, it causes this exact same effect. It causes 30S up and 21S down. And I showed that the miR28 siblings directly target RPS28 using UTR luciferase assays. And so, just to show you the same slide, depleting RPS28 alone essentially causes the exact same defect that treating cells with RPS28, miR28 or miR708 causes. There was other interesting things in that data. I didn't have time to explore all of it. We also did a miR-ECLIP experiment with Eclipse Lab Innovations in San Diego, California, and that showed us a whole other set of targets that these 2 microRNAs directly target. Austin has a question.
Yeah, so I have a question about the ribosomopathies that's always confused me. So a lot of them have very tissue-specific disease phenotype types. You you want to guess why?
So the field is already guessing why. There's two main camps. So Austin's question is, ribosomopathies have very tissue-specific defects, which I kind of glossed over today. Why is that? In other words, how could a defect in a protein that's in every single cell in your body cause tissue-specific defects? And so the field kind of has two ideas about that. Rachel Green in Baltimore believes that this may result from essentially tissue-specific defects, tissue-specific like changes in how much ribosome, how much ribosomal activity is needed, like in a specific tissue. Essentially, that's what her hypothesis is. And so that would mean that tissues like the brain, where probably a lot of things are going on during development, maybe you need a lot of proteins. If you don't have the right amount of ribosomes during development, maybe that's one reason that you could not be able to develop that.
Wouldn't that just argue that just the fastest proliferating cells are the cells that would care the most because they need to make the most?
Not necessarily. I think there's a correlation between certainly proliferation and translation, right? But it might, right? In development, you have all these timing things, right? This is what colleague Allison studies, right? That's what microRNAs control is built on timing. So something's wrong at the wrong time. It's not right at the right time. That could be a reason. The other compelling hypothesis is put forth by pre-rRNA and other people who believe in specialized ribosomes. I should say believe who are studying the possibility of specialized ribosomes. And in fact, they do have some compelling data. But essentially, a specialized ribosome is essentially what the hypothesis is that different tissues actually have slightly different makeups of their ribosome. And so the ribosome is like very well conserved, right? But it might be the case that for a certain ribosomal protein, there's actually different homologs that are very closely related. But perhaps if you have RPL37A versus RPL37B or whatever, that could actually change what transcripts are translated, right? Essentially the translation efficiency in that tissue. And so perhaps if you have, perhaps that would be a reason that those tissues would be specifically affected. But it's an outstanding, wonderful question in the field. Jake.
Yeah, so I'm going to take one of your questions on the slide. So
Thank
I was
you.
wondering if you can make protein with only rRNA in lab, and I'm going to add to this and ask why or why not.
What a great question. Well, I just so happen to have a slide to answer this. So I had to cut the slide, but it's really cool. And so in Adi Yonat's lab, who was one of Tom Steitz's co-laureates in the year 2000 for crystallizing, getting high resolution structures of the ribosome, they actually have been working on this project, I think, for a long time. And they published it last year in the Hour. And so what they found is you can take RNA only, essentially the fossilized cradle of the ribosome inside the ribosome, just rRNA, and you can use that to make proteins. And so what they found is that if you take just as RNA, which is essentially a dimer of essentially tRNA-like molecules, you can make protein. And so this top panel here is the 50S ribosome. So that's how you know this peak right here means the ribosome is making protein. In the negative control, where there's no activity, you're not making protein. But if you just feed it this RNA construct, it will actually make protein. And this is in vitro, I should say. So but you can use this RNA construct, and it will dimerize, just like the fossilized cradle of the ribosome, and it will make constructable, which is fascinating. The ribosome is a ribosome. Thank you for the question. Very cool stuff. Engineering mindset, by the way. Thank you for all the questions. Wait. Another question. Let's switch up. Congratulations. Thank you all. Thank you. Thanks everyone on Zoom. Okay. Any other questions? That's great. I have some informal acknowledgements now that I like to make. This is the appropriate. Okay, excellent. So, thank you all again so much for being here. I have just a few informal acknowledgements. Actually, my team will, so if you need to leave, please leave. Thank you for coming. Thank you so much for your support over all these years. Love all you guys. Okay. So of course I want to start with my fearless advisor, Susan Jacobsberger, MD, PhD. Yes, so Susan mentioned in her intro that she didn't know how I ended up in her lab, and I think it's some of it is must have been fate, right? Because actually Susan and I think alike on a lot of issues, and I'm so grateful that she's been such a trusting and guiding mentor to me in the lab. Susan has trusted me a lot to do crazy things like learn about microRNAs when neither of us knew anything about them, and to spend a lot of money on doing a screen and spend a lot more money on this assay and that assay. And to spend a lot of money on instruments, which I love buying because I'm an analytical chemist. And so we love instruments in the lab. But Susan, I'm such an improved scientist for being in your lab. Obviously my technical skills have improved greatly, but I'm such a better communicator because of you, whether on the page or on the stage. And I'm such a better philosopher of science because of you. And I want to thank you deeply for that. I also want to thank my committee. So thanks to Tony and Joan for being amazing scientists. All of you are very resilient scientists, I will say. I'm sure you've stood up to a review or two more times than you'd like to count. But I also admire you for being really resilient people, all three of you. And so thank you for being wonderful role models. I want to thank Jen Gurdon, my outside reader. Jen is a leader in our field and also in the field of chromosome biology more broadly. And she's awesome. I've met Jen a few times at conferences in person, and she's always a lovely person to talk to, and really I'm honored that she thinks critically about my work and has helped me with my work in my thesis more recently. Jen also wins the award for coolest CV line item, which is Guinness Book of World Records record for most complete genome. so Jen was on the team for the telomere to telomere consortium, which completed the human genome and actually illuminated what was once dark matter of the genome, the ribosomal DNA arrays, much to her delight as well as the rest of the field. So thank you, Jen. I also want to thank my other, all my other mentors. This is not all of them, but some of my other mentors that I could fit on one slide that have helped me get to the place that I am today. And I want to thank Chase Spurlock, who was a graduate student at Vanderbilt and let me come work in his lab when I was in high school, which is an extreme privilege. And he's doing wonderful things with machine learning now in Nashville. I want to thank Vitas and Don, who are my mentors, co-mentors at NIST. It's very exciting to work with both of them. I want to thank Dave Klippel, as well as Jens Mila and Alyssa Hare, who are my mentors at Vanderbilt. And I owe a huge debt of gratitude to Yulia and Laura at the YCMD. They also taught me so much about screening, as well as they put a lot of trust into me to operate their $1 million automated microscope by myself multiple times. And they showed me where the key was and everything. So that did wonders for my self-confidence. Science can often grind you down and make you feel like you don't know anything. But I sure know how to operate that microscope now. And I won't break it. So thank you so much, Julia and Laura. I want to thank all my lab members, past and present. So I can't fit everyone on the slide. That's Amber on the right there. And that was a box that we got an instrument in, and she was, for some reason, just chilling in there. So I had to check on her, do a wellness check, make sure she's okay. But all of my lab members, past and present, have been so supportive. This is one of the most, I think, supportive environments I think one could possibly do science in. Everyone is so together and supportive of each other and wants the best and success in lab and in life for each other. So it's truly an amazing opportunity to work with all of you in my career. I also want to thank my MB&B and Yale community. So thanks to everyone in my cohort, of course. I want to again shout out Mason, who we started together as other people in this room started with us, but we're ending together in the same lab, and it's been such an amazing opportunity and a joy to work with you over all these years. And you're a wonderful advocate and a true friend. So I thank you for that. And I want to thank all the other folks on these slides, everyone who's made my Yale and community experience what it is today. The other CB in our program, Carolyn Breckel, is attending right now, so I'm sure she's passed with flying colors. I also want to thank my New Haven community and Unite Here and New Haven Rising. It was truly the experience of a lifetime to work with you all, change Yale for the better, even just a little bit, and change the city and make a real impact here in our community. And I wouldn't have it any other way. So thank you so much for all you taught me about humanity and what it means to work together. Thank you. Of course, I would be remiss if I didn't acknowledge my beauties and beasts. So on the left here is Tipper. Tipper loves a carpet. This is Webster, who's in Susan's office right now. He loves peanut butter. And this is Flora, who loves being held, ideally in a bag. Again, this is Tipper. She loves a carpet. Webster loves his peanut butter. And Flora just loves being held in a bag, ideally. And one more time, Tipper loves a carpet. Webster was getting some peanut butter off screen, but he does also love to be in his Charlie Brown Great Pumpkin costume. And Flora loves being held. This is not in a bag, but she's in a costume, which I think she would count as close enough. Also, the Webster emoji pack is available for $2.99. So just message me if you want access to it. And I will do requests.
I need that.
Yeah, it's in the lab Slack, but I also I don't know if you can get custom ones. I want to thank my quote-unquote adopted family in Pennsylvania and other places around the world. So this young gentleman down here is named Dan Thompson. And I met Dan, I think, maybe a month into being here at Yale. He asked me a very strange question in the gym. But I said, I don't think that's what that is, but it's nice to meet you. My name is Carson, and we've become really, really close ever since. To the point where Dan's family, the Thompsons, have essentially, like I said, adopted Anthony and Sally as additional family members. And it's been so wonderful to make music with this whole crew, go around exploring, have some big life events we could celebrate, and be there actually for their family reunions, which is really special. And so thanks to everybody. Thank you, Dan, Chris, and Joel. Thank you, Wendy and Greg. And thank you to Uncle Steve and even Uncle Eric. And of course, I want to thank my blood family who's in the US and abroad. My family has been very supportive of me over all these years. My parents sacrificed a lot to provide all these opportunities for me. And I'm so grateful, and of course I can never repay them, but I do live every day and try to make them proud. I want to thank especially my mother who's here today, in the back, all the way from Rockvale, Tennessee. Mom, I want to thank you, of course, for being here today, but for always believing in me and going way out of your way to make sure I had all the opportunities that I could in my life. And you also taught me, actually from a very young age, about the real challenges and sexism and discrimination that you had faced and that other people face in the workplace. And I think that's informed a lot of my life, and I've certainly learned more as I've become less sheltered. But that was a really important lesson for me early in life, and I always want to carry that with me. I also want to thank my dad, Peter, who's at home right now with all his cats. My dad is incessantly curious. I think my mother would agree. He often asks me way too many questions and is annoying. But that's part of the reason that I'm a scientist. I got that gene from him. And he's also one of the kindest and most giving people I think I've ever met in my life. And that's the lesson that I'm trying to carry forward to everybody in my life that I come into contact with. It's always a good day when you can help somebody out. And so I thank you for teaching me that. And I think our French connection, our French relatives here, I want to thank Bibi and Tab, my aunt and my uncle, as he likes to call himself, my uncle, and our relatives in California, my cousins. And I want to also thank Sally's family for tuning in. And of course, I have to thank my wife, my beautiful wife. Yeah, so I am making fun of my wife a little bit here. But she's an incredible person. She brings so much joy and support to me in my life. there's not really much that you can really say about somebody to as important as your spouse. Right? I know, Sally, I love you so much. And this is what Sally would be if she got engaged. Dr. Snack, she loves snacks. And she's always doing some kind of crazy crap in her house that may set the fire alarm. Okay. So now we can let them eat cake and it's a chip to you. Thank you so much for coming. Let's go eat cake. So we're having a reception in just down the hall, 125. Carrot cake from Claire's Cornucopia, some other things to snack on. Plenty of drinks and beverages. Please help yourself. I'm going to have some water and I'll join you. Thank you all so much.
Yeah, I'm gonna stop by my lab and send my show.
What does it say? Carson's defense. What's the other Carson? Oh, I love it. Wow.
Yeah, they're a little expensive, but would you like to
Ooh, that won't fit me. That will fit you. Are you kidding?
I brought a bigger one.
But I do need the Webster emoji pack.
So here's what I do need.
Wasn't that cute? He's so cute.
Great stuff, Carson.
Thanks, John. And now you'll never forget that the ribosome is a ribosome.
Wait a minute. You can set a